and welcome to the Orkney Science Festival. My name is Beth Ansell and I will be introducing this fascinating talk on the human genome and DNA sequencing. Now, DNA sequencing may not be a familiar topic to some of us, but it's actually happening across Orkney as part of the Orchids Health Study. So what is a genome? What is DNA sequencing? And why is it important? Here to answer these questions is Dr. Lucija Kalrić. Dr. Kalrić is a UK Research and Innovation Fellow at the Medical Research Council's Institute of Genetics and Molecular Medicine. She comes from Croatia, but currently lives in Scotland. Before we begin, fire exits are through this door, through that door, and through the back door. So let's start by giving a huge welcome to our speaker today, Dr. Lucija Kalrić.
Hi, good afternoon everyone. Thank you for coming in such great numbers. I'm Lucija, and thank you Beth for a nice introduction. I'm coming from Croatia originally, but I now live in Scotland. And my current research area of interest is whole genome sequencing. So when I talk to my parents about what I do for work, I often hear from my dad, oh yeah, no, but it's obvious. You got your looks from your mom and your smarts from me, of course. Then I go to my mom and I ask her, so what do you think? And she says, no, completely opposite. You get everything from me, both looks. But in reality, of course, we get half of our DNA from our parents and the other half from my mother and the other half from my father. I often find myself saying, oh yeah, you know, I like eating chocolate so much, but it's in my genes, you know. I can't help it, it's just running in my genes. And I assume how many of you here had said something similar in the past? Yeah, you all do that, but how often do you stop and ask, what does it actually mean? It's running in my genes. Okay, I got it from my parents, but how does it actually express itself in a way where I can say, yes, chocolate is very nice. So I hope that today I'll show you what a DNA is. We'll learn something about the basic structure of the DNA, and also show some examples how it might influence our visible traits and how it might have influence on our disease, on different diseases. So I start with what was potentially a heartbreaking story, but turned out as a great success in the end. So this little boy here, in 2009, was only 6 years old, and by that time he already spent more than 70 days in a hospital, so that's a third of his life. And he already went through more than 100 surgeries. He had a very serious gut disease, very rare, and doctors had no idea what was happening. So they did everything they could, trying to help him, used all the currently available diagnostics, but there was simply no answers. And then, at some point, partly because of persistence of his mother, they finally had his DNA sequenced. And sequencing his DNA helped finding what's really the cause of his disease. And his disease was whatever he would eat, he would get instant gut inflammation, and small holes would start forming, poking holes in his intestines. And as soon as they knew the cause, they could treat, they could act accordingly. And he was lucky that there was a way to help with his diagnosis, and now he's a relatively healthy 15-year-old boy. So this is one of the first success stories of the whole genome sequencing. But let's go to the beginning. Let's go all the way back to 1953, when Franklin, Crick, and Watson discovered the structure of DNA. This is now something that we are all well aware of. We all know that when I say DNA, we all think of this double-stranded helix. But in 1953, this was a really big discovery. And this beautifully simple molecule is actually very, very simple. So it's built of only 4 building blocks. We call them nucleotides. And there's one called adenine, thymine, guanine, and cytosine. So there are just 4 different molecules. And for the simplicity, we tend to call them just by their first letters, A, T, G, and C. A usually binds with T, in the opposite chains, G with C. And the way how to remember this, found on the internet, is, I heard an acronym for, I like looking at George Clooney. I'm not sure how true it is, but anyways, it's a neat way to remember, regardless of what you think of George Clooney. And so these nucleotides, they bind one by another and form this long molecule, which has two strands. So this is one strand, and on the opposite side, there's another strand. So one molecule of DNA consists of two strands, and so this is where we see in the different strands, this is where this pairing George Clooney happens. What's also important for future slides, that base pair is just like this one building block of the both chains. I'll mention that in the future. So all this molecule, they bind one for another and form this very long and double helix structure. Now, if I already said that we know what these basic building blocks are, so you and I here can now sequence this small molecule of DNA. What sequencing means, it means just reading out what these letters, what these nucleotides are. So I already said that what's in green, that's a guanine. Alright, the next one is A, the next one is again G, and the next one is A. And on the other side we have cytosine, thymine, cytosine, thymine. So what we just did here is a very simple way of, we just sequenced this small molecule of DNA. And the whole genome is a complete DNA sequence of an organism. So reading out all these letters of one organism. So in principle, the idea behind it is very simple, but causes and difference contribute to all the variety we see between the organisms and between each other. So the human genome, our whole sequence, consists of 3.6 billion base pairs, so these building blocks. That's 2 times 3. I'm really bad with big numbers, I just don't understand what a billion is. So if I ask you, spoiler, so if I ask you, if I would print out a sequence, all these 3 billion base pairs, how many books do you think that it would be? Would it be one book? Would it be one big book? Several books? Answer is a lot. So this is a printout of the first human genome sequence, and I think, it starts with a 2, so 1 is missing. So it's even more than what's here. So it's inconceivably big. Now let's think about it in the sense of size. So we have 3.6 billion base pairs, all right? One base pair, if I would measure the size of this, is 340 picometers. I'm also very bad with small numbers. So that's 12 zeros after the first 0. It seems very small, but this is very big. So if you would multiply one with another, you would get the total length of human DNA, and it comes up to 2 meters. So if you would put one right next to each other, you would get 2 meters long molecule. And every cell in your body has 2 meters of DNA. And you may wonder, how is that possible? So I have a small example. So let's imagine that this is this is 40 centimeters molecule of DNA. And if I would put it just like this, of course it wouldn't fit in the cell. But what happens, this DNA starts folding and folds, and you can already see what's happening. Just by the way it's folding, is reducing in its size, and it gets more and more complex, and then more and more complex, and in that way it forms a chromosome. You all heard about a chromosome. And that way we can fit 23 chromosomes that we have, 46, half from mother, half from father, in all the cells that we have. Now, pop quiz time. So let's imagine that we whole genome sequenced these different organisms. So we have a human, we have a mouse, we have a monkey, we have a wheat, and we have a bacteria. And we determined the sequence, we know how long, how many base pairs all these sequences have. So my question to you now is, how would you order them based on the size? So A, do you think it's exactly in the way it's ordered here? B, monkey's genome is bigger than mouse genome. C, bacteria has the biggest genome. Or D, wheat has the biggest genome. So who's up for A? All right. B? Is it just because since I put it in this order, there must be something that's not right? C? All right. D? Wow, this is a very good public audience. Very well. The correct answer is D. So wheat has almost 3.5 times more base pairs than a human. And the only thing this is just saying that the size of the genome does not say nothing about its complexity. So both us and mouse and a monkey have more or less similar number of nucleotides. Okay, so now we learned what the DNA structure is. The next question, why is it important? So DNA is a cookbook for proteins, and proteins are those that have a role in every aspect of your life, of every part of your cell biology. They have a role in everything. For example, proteins have a structural role, keratin, so your hair is built from the keratin. They have a function in defense, so when you get infected by bacteria or something from the outside, antibodies, another proteins react to prevent infection. They have a role in transport. You've probably heard about hemoglobin, so that's a protein that carries oxygen in the red blood of your cells throughout the body, making sure that we get oxygen in all the cells. They have a role in signaling. There are receptors on cell membranes. So, for example, when you're in a dangerous situation, adrenaline hormone will sit on these receptors and activate needed response, for example, run away or whatever is needed at the time. They also have a role in different chemical reactions, so they help digest your food. They help do all the chemical reactions that happen in your body. So proteins we know that are important, and we know which proteins, we build proteins based on our instructions in our DNA. How does it actually work? So I said it's a cookbook for proteins. Proteins are built from another building blocks called amino acids. And amino acids are basically, we know in a protein which amino acid to put in which position based on this translational code. So it works in a way that you have three DNA letters, one into another, and they one next to each other, and they define a certain amino acid. We'll look at an example now. So for example, let's imagine this is a DNA molecule. What happens, you read 3 by 3 letters, and each of these 3 letters define a different amino acid. So this three-letter codon defines threonine amino acid, next one lysine, next one glutamine, and the next one glutamine. So what happens, our DNA is read out 3 letters at a time, and specific amino acids are added one after another until they form a whole protein. So it goes on for a bit longer than just 4. So then you get a big complex molecule protein. This one is specifically hemoglobin, I already quickly mentioned. This is a molecule that has ability to bind oxygen on this in 4 different positions in its structure. And it's important because they are inside of our erythrocytes, red blood cells, and they're in our bloodstream and make sure that all the cells in our body get enough oxygen at the time when they need it. So now the question is, so this is a whole DNA sequence, but at least part of it is not whole, of this protein, which gets translated 3 by 3 letters to form a protein. So the question is, what happens if I would change one of these letters? If I would have a change here, for example. What would happen, and what sometimes can happen, that just a single letter change, which we then call a single nucleotide polymorphism, because it's one nucleotide, it can happen that it results in a completely different amino acid. So in this case, this was the old sequence we had. We had threonine, lysine, glutamine, glutamine. If we change the sequence that here, instead of A, we have a T, we could have a completely different, not completely, we get one different amino acid. So now instead of threonine, lysine, glutamine, we have valine, and then again glutamine. And this sometimes can have very serious consequences, And this is a real example from what happens in sickle cell anemia. So when you have hemoglobin that has this change, amino acid, your red blood cells are no longer this nicely roundly shaped. They become this arrowed and this coma-looking like cells. They have shorter lifespan, and the other problem that happens, the erythrocytes roll in your veins, and when you no longer have a proper nice round shape, they tend to stuck and they tend to make clots. And that was just a single letter that was changed in the sequence of this protein. Of course, that's what happens when you have an extreme case. But we all know that my height, we all know that all of our traits that we see are defined by our genes, so there must be something more behind it. For example, if you take an example of height, on this side, you have a height of parents, so average height of mother and father, and on this side, we have an average height of a child. So just from the height of parent, you can estimate, you can guess more or less precisely how tall your child will be. And of course, since it's inherited from a parent, it also has to be somehow governed by DNA. And this is a different example. So what happens when we have a variation, how some of these more complex traits are regulated? So now I'm representing our 2 meters long string of DNA, spoiler again, in this just consecutive line. And you see, you don't see, but different colors are different chromosomes. And this is all the different regions of these chromosomes that are involved in the regulation of height. So these are all the different positions in your, let's say, in your different genes that have some role in defining how tall you are. So in some cases, we get single change that results in more or less catastrophe, having a different protein whose function no longer works. In some other cases, in most other cases in healthy human beings, we have many different positions in DNA, many different genes that are involved in defining our visible traits. Okay, sum up. So variation DNA can influence our traits in a way that it changes the protein structure, so changing the hemoglobin. Or, I didn't show this, but another way, simplified, is it can also change the amount of protein. Sometimes it's not just what's the form of the protein, sometimes it's also about how much protein you have. So next question you could ask is, how do we learn which part of DNA influences which trait? Well, let's take this simple example. Let's take this few giraffes, and let's assume that they are all adults, so there's no baby giraffes here. And let's assume they're all of the same gender. We know that female and men are usually of different height. And now let's ask this one question. So let's take 2 of these single nucleotide polymorphisms in a DNA, and let's just look which letters these giraffes had. So we took this, we call this single nucleotide polymorphisms with certain IDs, so we know what we are talking about between each other. But let's say we genotype this, we define the letters these giraffes have, and just group them based on their letter. So on this side you can see these giraffes have TT, TC, CC, CC, so one is coming from mother, one is coming from father, that's why I have 2. Do we see any trend? Not really. Okay, so then in that case you would say this position in DNA has nothing to do with height. In the next example, you can guess how this one will end. We genotype them again on a different part of the DNA, and we got this. So now here we see AA short, AG halfway through, GG tall. So now we see that this position in DNA, depending on which nucleotides you'll have a different height. And we do this, so we start with this, when we have a new trait, we start with this. So this is the whole genome, well, not 3.6 billion base pairs, but some of them. And we ask the same question. If we group giraffes based on their letters, will they be tall or not? And in the case of height, we get this. So now behind these guys, all this if you were here on the previous talk, this is called a Manhattan plot. Behind each of these pillars, there's a gene that's somehow related with our trait of interest, and now we can take the list of these genes and try to figure out what's actually happening behind it. Okay, so recap, DNA is built of letters, A, T, G, and C, letters, nucleotides. Sequencing is just reading out the exact order of these letters. DNA is a recipe for a protein, and we have a change in the DNA, variation DNA sequence can result in a serious disease, in some cases, in rare cases, or in most cases it results in the variation in all the traits, all the different traits we have here in this room. Eye color, hair color, everything else. Okay, so that was a brief recap of what should help us with our travel back in future. But we have to go back to the past to the 1990, when one of the most important projects at least in genetics happened. Human Genome Project was started by US Department of Energy and National Institute of Health, and the project was trying to find a genetic blueprint for building a human being. So they wanted to take person and sequence and learn exact order of all the nucleotides in their sequence. Goals were identify all the genes in the human DNA, determine the exact sequence, and develop tools which would help that we can use this in future for potentially diagnostics and future research. In 2000, 10 years after, Bill Clinton announced that they are here to celebrate the completion of the first survey of the entire human genome. And he said that without a doubt, this is the most important, most wondrous map ever produced by humankind. That's quite a strong statement. Slight problem, this was in June 2000. The DNA sequence first draft was released only in 2001, but the elections were in 2000, so they had to, it was 90% finished by this point. So what were the initial findings? So their initial findings in 2001 and then later in 2003 were identify all genes in human DNA. So they found that there's approximately 22,000 protein coding genes, so those that will form a protein. They determined the exact sequence, so we have a sequence now, but they also learned that the structure is more complex than I expected. And they developed the tools, but it was just a good start. It required a lot of research to get to this point, but that enabled development of many other methodologies that followed. You may wonder why. Why is it just our egoists, we are humans and we are better than the rest of the world? Why do we need to know our DNA sequence? But it's quite important for our well-being. If we define a reference sequence of an average healthy person, we can use this sequence to help to learn something about diseases. For example, if I have a healthy individual whose DNA sequence is like this, and then I have a patient, and I'm not sure what's wrong with the patient, as in the case of the Nick Walter, I could compare his sequence to the healthy sequence, and I can find, oh yes, this patient here has a different letter, maybe that's the reason why he's not feeling well. And this, finding out where this is and which genes are involved, can help us develop new future drug targets. So if we know what's wrong, we can try acting on it. And that's exactly what happened with Nicholas Walker. So what they discovered when they did this first, when they sequenced his genome, they discovered that in one position in DNA, he has, instead of guanine, he has alanine, and that changes to completely, tyrosine replaces cysteine in this gene called XIAP, and that's what's causing him all the trouble. This gene here is involved in the control of inflammation in the immune cells, and with this knowledge, I mean, this is a rare example, and this happens when you have a serious rare disease. You're more likely to have such finding, but in a way they were lucky because they had this relatively simple case, and it was just one letter in one gene that had the most sense. So knowing this, he got a bone marrow transplant, so he changed all the immune cells in his body, and now he is relatively fine. He still feels some consequences of the first 6 years when they were trying to figure out what was happening, so some damage was already introduced, but if they did this earlier, he might have been completely fine by now. So that's one reason why we want to have a reference sequence. The other reason is, can we also use it to predict the drug response? So for example, if we have a group of patients with the same condition, can we sequence them and then see, and of course, same condition that are treated with the same medicine, and you know some react and some don't. Well, let's try sequencing them and seeing if those that react to, respond to the drug, have a different genotype, different letters than those who don't. And if we are lucky enough to find the difference, for the future patient, we can first assess, would this patient respond to drug or no, or not? Because some of these drugs have very adverse effects, so you would like to prevent giving something that probably has serious complications than others. And this is something so this is a study from 2017. So this is no longer a whole genome sequencing. When you know which gene is needed, you just focus on that gene, and you just sequence that gene, not the whole genome. These guys here, they took 137 FDA-approved drugs that existed by 2016 at the American market. These were the drugs that had a pharmacogenetics label with them, saying that there's some support, at least some support, that some gene is related with the metabolism of the drug. They had a very number of criteria to reduce the list, so they ended up only with 10 drugs in the study, and in their study, but they took economic evaluations from all the other studies. So there was up to 20 studies testing if we test for genetic predisposition for drug response and give appropriate drug, will that reduce or increase cost of the medical treatment? And this is a summary pie chart. So green, as you would expect, means it was either cost effective. Cost effective means it did come with an extra cost, but this cost was worth it because it either improved the quality of life in the future or it really helped with the diagnosis. And this part is cost saving. So this part it was actually cheaper to do this than try testing with many different drugs and so on. And red it's not cost effective, and this yellowish is they couldn't determine. And so here you have the list of these drugs that they tested. And these are all the drugs that have implications that there probably is a gene we could test for. Warfarin, this one is involved in blood clotting. I don't know all of them. For example, this is AIDS, HIV drug, and so on. And so, in summary, they could see that at least for 57% of these drugs, it might be worth checking if the person will respond to the drug or not. Okay, so the Human Genome Project was announced finished in 2003, and whoever is interested, you can just download the reference genome. It's freely available. You can print it out if you want. It's probably pointless. You can try reading it. It's also probably pointless. But there are other databases that can help you to interpret what you are seeing. So there, of course, now there's a big effort in trying to understand what the whole sequence actually means. Important thing, the total cost of this Human Genome Project was $2.7 billion, or that's in 2003. That's around $5 billion with the inflation in the between time. Of course, that was the whole development, everything. At the end of it, sequencing of one sample was around $100 million. So that's quite pricey, and we probably can't afford that on a high scale. So that's when we came to the next question. So whole genome sequencing gives you a completely crisp picture of your genome. So this is, I'm from Croatia, so this is Dubrovnik. Recommended to see it off-season. From this picture, you can see everything. You can see there are boats. You can see that the weather is a little cloudy. It's been dry for a while because the leaves are yellow. There are not that many tourists, so it's probably May. So we can see many things. We can see there's sea. It's a town on the sea. It has trees. It has boats and so on. But we can go into much further details. And this is what the whole genome sequencing gives us. We can ask, what if we take something in small resolution but at lower cost? So can we do something that we still see all the main features? It's sea, it's a city, there's some trees, there's some boats. So you could still learn something from this, but at much lower cost. And that's exactly what the next project did, the International HeartMap project. It started in 2002. They said, okay, it's very expensive to sequence every base pair. Let's try sequencing every one in 5,000 in the first phase, and then went higher resolution in the next phase. And they took 30 people from different populations in the world, assuming we are different, so there must be some population-specific differences. So they took samples from Nigeria, equivalent of Europe, China, and Japan. And what this did, so this was much cheaper. This was orders of magnitude cheaper. So what this resulted in that since 2008, well, even before that, it started in first successful study was in around, I think, 2007. But we had this big boom in discoveries of genetics-related discoveries with diseases and complex traits. What this graph shows, first, this is the cumulative number, so the summed up number of all the different regions of DNA, all of the different single nucleotide polymorphisms related with the disease. And in the circles you can see the number of new discoveries in that year, and the colorful figures on the side are all the different diseases and traits that were tested and that had a genetic, that were involved in the genetic study. So you could see that this enabled a huge increase in discoveries. But now, of course, at the same time, they continued working on how to make the whole genome sequencing cheaper, because in some cases we really do want to know if the leaves dried because it was too dry for too long. So in 2001, the whole genome sequence was $100 million, and in 2017 it fell down to more or less 1,000. Of course, at the same time, this fell down to more or less, I think, 200. Cheap, but still this is much cheaper than where we started off. And now this enables us to do big population studies, as the one that was done here in Orkney, Orkney Complex Disease Study, and the big Viking Health Study, Shetland. And so I'll show you now one example from these studies. We have whole genome sequencing for relatively short, so we don't still have many things to say, but we have one example where it worked very well and why we think it's very important to continue working on it. So in these studies, we have CRISPR-clear picture of whole genome sequencing for 1,800 people. We have this SNP chip, this low resolution but cheaper version for 4,200 people approximately. And we have something else. So this is called whole exome sequencing. I didn't mention this before, but this is just looking at these parts of DNA that are defining proteins rather than the whole sequence, because most of the sequence is not defining proteins, but it's telling us more on how to regulate these proteins. This is just looking at a protein, so it means things that are more likely to have more serious implications sooner. So it's just giving us part of the picture, but at a CRISPR-clear resolution. And by now, until now, mostly coming from this part, these studies contributed to the discovery of more than 100 novel associations of different traits, different diseases, and so on. But I'll now follow to a specific example from the Viking Health Study. So this is a study that started in 2013 and finished in 2015 in Shetland. To be able to take part in the study, you had to have 3 or 4 grandparents from Shetland. I think for more than 3 or 4. So you had to have a heritage from the islands. And we had 500 people their whole genome sequenced, and around 2,000 had a SNP chip, so this low resolution picture. So we had one instance of, we call it a clinically actionable disease. So this is something that can have a example similar to hemoglobin and NIC, maybe not at this scale, but something that we know if you have a variant, you should probably go to visit your GP and act accordingly. It's not something that you will necessarily impact you right away, but it's something that probably needs some addressing. So we know that within this 500 people, we found that one person had a polymorphism that's related with our trait of interest. But we have only 500, but we have 2,000 people more in the 150,000 people, 1,500 people more in the study. So what we did was the following. We reconstructed the family tree of this person, and we asked, so we know we don't need to whole genome sequence, we can now take this specific region, and we looked at this specific region of 42 of his other family members, just the region of his, and discovered we find 2 other individuals that were carrying the same SNP. So this is just in this family. Now the question is there anyone else in the population that's not necessarily close related to this family that has the same? And now we reverted to a small trick to use this low resolution data. So we did the following. So we know 3 individuals that were affected, and this is a way of visualizing this part of their DNA. So this is 1 person, 2nd person, 3rd person, so each column is one person. And as I said, you have 1 chromosome from your dad, 1 from your mom, so each of these 2, one is coming from one parent, other is coming from the other parent, let's say. And we just tried to find out these 3 people. This is the region where we know the variant should be. Is there something similar in these sequences? And I don't know if you can see it. Yeah, okay, I helped you. If you look at it, these is exactly the same as this one and this one. So now we could define, I color-coded this as 0s and 1s because that's the way of easier working with it. It's a DNA sequence behind it. So define this sequence, so we now know that this is the sequence that individuals that had the variant, this is how their sequence surrounding the variant looks like. And then we ask, okay, we have remaining 1,500 people. Can we look for the same sequence in these other people? And we did that and searched through the remaining 1,500 samples. And indeed, we find 2 additional persons that carried the same sequence surrounding the variant of interest. We went again and just took these individuals and just sequenced this part of the genome, and we confirmed indeed they do carry this variant. We reported that this, So in this way we use the combination, I mean it's still $1,000 is much less than 100 million, but it's still, if you want to do it 2,000 times, it's again the big numbers problem. So we have to find ways where we can combine different data we have to try to get to the same information. Okay, so this is one example of what we can find, what we found already in these data, and I guess there are more to follow as we investigate more. So, to summarize it all in a way, what would happen if I would have my own whole genome sequenced? So I'm a relatively healthy person, relatively young as well, so I don't have any obvious disease. So what would happen in my case? I could print out my book. I live in a small flat, so that would really make no sense at all. I could learn what are my predispositions for diseases. For example, I could learn that I have 3 times higher risk for dementia. I didn't sequence, so these are made-up numbers. Well, not made-up numbers, it's a made-up example. But what does it mean? Should I go to the doctor right away? Probably yes. Should I get stressed? Not really, because 3 times higher risk for dementia. The chance of getting dementia is, I think, around 5% during the older you are, the higher chance you have. So my risk will now increase. I have 15% chance of having a dementia. If I ask you, tomorrow there's 50% chance there will be sunshine, will you leave your waterproofs home? Probably no, because I mean, you'll get soaked, more or less. The advice is, yes, I should probably go regularly to my doctor. I should, especially as I get older, I should keep an eye on if I'm having any symptoms, if I can remember which they are. And I can notify my family to maybe do so as well, because they might be more helpful in this case. Yeah, so regular checkups. So I can potentially change my diet to help reduce some environmental risk and things like that. I mean, it ends up in we spend billions and billions to learn the same. Probably eat less and move more. But still. The other thing in the future, if I get any disease, that there's a drug for it, and we know that there might be a genetics-related response to the drug, my doctors could go back and check if they should give me a drug or not. And sorry, this is a spelling mistake. But one other thing that I also learned, but we rarely think about, I will learn much about my family. Because I'm inheriting half from my father, half from my mother, and I have siblings. So my knowledge will also affect them. This is something that's a completely different talk to give. And the question is, I want to know this information. Do my parents want to know as well? Okay, so I guess this was a brief introduction of what a whole genome sequencing is. I hope we learned what a DNA structure is, how it can influence our traits, and I hope we got a just small peek in what the future might hold. Thank you for your attention.